Hey guys, this is Shelby with the Rochester Public Library. So this week for Full Steam Ahead, I wanted to talk to you guys about biotechnology. And what biotechnology is, it's basically using living organisms or biological processes to make products, right? So when we talked last week about domestication, we can kind of think of that as almost the earliest form of biotechnology, right? We're taking organisms that we find in the world, like grasses or flowers, and we're modifying them using artificial selection to make them more useful or human based, turning them into products. And there's another kind of biotechnology as well that kind of developed alongside domestication, and that's kind of the basis for our more modern biotechnology that we use today, and that's fermentation, right? So fermentation is basically the process of exposing foods to microorganisms like yeast or lactobacilli, which are a special kind of bacteria. And these different microorganisms, instead of spoiling the food like normally happens, instead they produce different chemicals that preserve the food. And these are microorganisms that are present basically everywhere, right? There's probably a lot of yeast and stuff floating around inside this room. And so these aren't, to some extent, they're natural processes, but they're also processes that humans have deliberately taken advantage of and used and developed to make food tastier and make it last longer. And so for our experiments this week, we're going to make bread, right? So what we have here is warm tap water. It's a 1/4 cup, and I added two cubes of sugar to it, so that's about 2 teaspoons. I'm also going to be using yeast, right? This is baker's yeast, Saccharomyces cerevisiae. We've looked at this a little bit before when we talked about fungi. This pouch has about 2 1/4 teaspoons in it, but it's not, you don't need exactly that amount, right? It's just right, so we're going to add it to here. And what this does is, the yeast is a living organism, and what this does is it wakes it up, right? So I can already smell it giving off nice tasty fumes. The yeast, they're fermenting, right? They're consuming the sugars that we put in the water, and they're using that to bud and to grow, right? And then in this bowl over here, we're going to add about a cup of flour. We might need more, but this is a good start. We also have a little bit of extra water over here in case we need it. Right, so we have our flour, and we're going to add a little bit of salt. Right, this is still going. We can let it sit for a while. Right, so bread is actually like super old, right? We actually have archaeological evidence from things like pottery and campfires of people making bread as far or as long ago as 14,000 years. So like before we even started domesticating wheat and rye and other things that we use to make bread, right? But this bread was probably unleavened. It was a flat bread, kind of like a tortilla or a cracker. And leavened bread, the kind that you use yeast in to make it rise and make it very airy, like the bread that we eat, the like loaves of bread that you get from the store, right? That's newer. We think that it originated in ancient Egypt, right? So here's this is from a tomb in Luxor, which is an ancient Egyptian sitting on the Nile, right? And this is a diagram of people making bread, right? But when the Egyptians were first making leavened bread, they didn't have this kind of yeast, right? They had to use wild yeast, right? And what they do is basically just leave the dough out, and things start growing in it. Yeast, but on top of yeast, also the lactobacilli. And so instead of this kind of, I don't even know what you call it, this is kind of a more bready taste. Whereas there would have been a lot of different flavors and stuff in Egyptian bread. It would have been more closer to sourdough bread, if you've ever had that before. Right, and the other way that they would have made bread is by using yeast that they got from making things like mead. Right, so we're going to add the nice yeast water in here now. And you're going to stir it up. Okay. We will need a little bit more water. We're just trying to add enough water so that it makes kind of a nice dough that we can pick up with our hands. Let's see. All right, so it's kind of falling apart still. That means it needs more water. So this is the easy part, right? We have the dough, and we just need to knead it. And you can do this for as long as you want. There's not an exact time, right? This is an experiment, right? You're trying to varying the levels of water and flour in the kneading time and seeing, and the bread is going to be different. It's not going to be exactly the same each time you make it, just based on these different variables that you change. Get a little bit more flour. Okay, so once it's like this, you're gonna set it in a pan, and then this is the hard part, is you have to wait, because the fermentation, it doesn't happen all at once. It's a gradual process. So what you leave the dough in a nice warm place, and you just let it sit for about an hour, and then you'll want to knead it again and put it in a baking pan. Like that, we're kind of taking a step out. It's already in the baking pan. And then you let it sit for another 30 minutes. And with the grown-ups' help, you put it in an oven. I used the same recipe before, or when I used the same recipe before, I baked it at 350 degrees Fahrenheit for about 40 minutes, and it turned out really nice. Right, but it is you'll want to check on it. And because you're using an oven, like I said, you need a grown-up's help. This is some bread that I made this past weekend. Pretty similar. I added a couple different things, right? So this one has a little bit of olive oil in it. It also has cornmeal, so it's a little bit of a different texture. But other than that, it's basically the same thing, same recipe. So fermentation is not just used to make bread. It actually it's used to make a lot of different foods, as well as important chemicals, right? So for instance, vinegar is produced using fermentation. So are things like cheese and pickles and soy sauce. And there's also a lot of really useful chemicals that we produce this way, right? So one of them is citric acid, which isn't a food, but it's kind of a food additive. Another one is ethanol, which has a lot of different uses as like a fuel and other things. And even though we've been doing fermentation for a long time, though, our scientific knowledge of it is newish, right? Really, the first person to describe fermentation scientifically was Louis Pasteur in the 1850s. And once he did that, it kind of set off a chain of events where people started doing fermentation industrially and started producing, this like dried baker's cheese that you can get at the store now, right? And that's the rise of industrial fermentation is really like one of the main legs of biotechnology, and the other one is artificial selection, right? Genetic modification is another way to put it. And for a really, really long time, the only way that we could genetically modify organisms through selective breeding, right? So breeding together different types of corn or different types of tomatoes to get one that we liked. And that changed in the 20th century, right? So the first new way of genetic modification we developed is called mutagenesis. And basically what this is you take some seeds and you would expose them to radiation, right? Like nuclear radiation. And what this would do is it would make the seeds mutate. And sometimes these mutations would be good. You'd end up with some really cool new plants that you could sell to farmers and they could grow them, and everyone was happy. But more often, the mutations would be harmful and it would kind of be a dead end, right? So this wasn't a really super efficient way of genetic modification. And then once we figured out the structure of DNA in 1953, we got a little bit more sophisticated and developed something called transgenesis. And what transgenesis is taking a segment of DNA, a gene, from one organism and putting it into another. And there's a couple different ways to do this, right? But the easiest way is using bacteria, right? You basically, because bacteria already exchange genes with each other, right? They have plasmids, just chunks of DNA that pass between different cells, in what's called horizontal gene transfer. And we use this kind of genetic modification to make like special kinds of yeast, right? Scientists made genetically modified yeast, the same organism that's in this, to produce something called insulin, which is a really, really important medicine that millions of people use every day to treat a condition called diabetes, right? And when you genetically modify a single cell organism this way, it's relatively simple. But it gets a lot more complex when you start talking about multicellular organisms like plants. And this process can take up a really long time, up to 15 years, and involve really complex equipment like things called gene guns, which don't shoot bullets, they shoot actually particles of DNA in gold at cells until it gets inside. Or even bacteria, right? The special bacteria that's been modified to make the plants grow different types of chemicals and stuff. And then there's kind of been a new development in genetic modification over the past 10 years, and this is called CRISPR. This is what it looks like. This is a type of CRISPR called Cas9, and it's basically a really weird, funky protein. And what CRISPR stands for is clustered, regularly interspaced, short palindromic repeats. So it's a bit like an immune system for bacteria, right? So bacteria have predators, right? So you think about like some animals eat other animals, where there's viruses that eat bacteria. They're called bacteriophages. And even when bacteriophages don't destroy a bacterium, they will change their DNA, right? And that's not normally a good thing. So, and what CRISPR does is it's kind of like an undo key, or like a save file. And it makes it so it'll find the changes that viruses have made in a bacterium's DNA, and it'll revert it to what it was before. And we can use this technology to modify basically really anything, right? You can use it to modify the DNA of a fungus or a plant or an animal, and it makes the genetic modification a lot faster and a lot cheaper, so that people can make or genetically modify things in months instead of years or decades. And these are the two ladies who kind of developed this technology. So this is Emmanuelle Charpentier and Jennifer Doudna. And they actually won the Nobel Prize for Medicine last year for the work that they did on CRISPR. So far we've been talking about genetically modifying plants and animals, but you can also genetically modify humans, right? This is sometimes called human gene editing, and it's a really, really controversial topic, right? for not really scientific reasons. We know how to do it, but there's a lot of really big ethical questions that we need to consider. And there are a lot of restrictions on the type of human gene editing that are allowed, right? So that scientists are only allowed to make changes that won't be inherited, right? So what they do is they change the somatic cells instead of the germline cells in a person. And that means that if something goes wrong, if the experiment harms someone, it won't affect that person's children if they have them later on. And as far as I know, there's only been one instance of human germline modification, and that was in 2018 in China. And the scientist who did this got in a lot of trouble, and he actually went to jail because he wasn't supposed to do it. But that's kind of another topic and a little bit broader and more complicated than we can get in here. So instead, we're going to do a really fun craft, right? And this is a corn cob that I made using pony beads. And corn is kind of at the heart of genetic modification, right? Most corn grown in the U.S. is genetically modified, normally to produce insecticides or to be resistant to herbicides, just so that farmers have to use those chemicals less and they can get more crops. And they're also used as what's called a feedstock, right? So when citric acid is produced, you start off with something called corn steep liquid, which is kind of like a leftover thing when you process corn. And that's what gets turned into this. But to make these, you're going to take 4 pipe cleaners. You're going to kind of fold them together like this. That they make, I guess you could call it a spider. Okay, so it's kind of like a little X, and you fold them up, and this is the basic shape, right? And then that takes like 30 seconds to make, and then the hard part, the part that takes a long time, is putting the beads on it. So you do basically just go like this. Push them over and they fit right on really easily. Ta-da. When I made this one, I think I put about 15 beads on each strand, and then I just twist them together at the top like that, and then you will end up with a little basic corncob shape. It's pretty easy to do. You can. It's just a little bit tedious. It will take a while to put all the beads on there, just because you have to use so many. But that's what I have for you guys today. And this is the last full steam ad for a while. We're gonna take a little bit of a break, and then we'll be back with more videos in June. So I'll see you then.